Hi everyone. So this is going to be the video that we missed due to my power outage on Wednesday. The focus of this video is going to be on primary, secondary, tertiary, and quaternary structures of proteins. And then after that, I'll make a supplementary video about enzymes. So where we left off on Monday, right? That would be Monday, was forming peptide bonds, right? And that's what we see in this picture to the right, in which two amino acids can be joined through the C terminus of one amino acid and the N terminus of another to form that peptide or that amide bond, right? And we've already discussed that proteins are sequences of these amino acids, and they can be as short as, depends. We talk about peptides. Peptides can be from 2 to around 50. It's a gray zone. Beyond 50, we usually term them proteins. And proteins can go from 50 up to hundreds of amino acids in length. And so we'll start to talk about how structure forms, what are the structures, and then when we get into enzymes, we'll talk about how structures can affect reactivity. So what we'll start with first is primary structure. And if it will let me change slides, there we go. And so this is what we've already talked about. So primary structure starts, literally, if to boil it down as simply as it is, it's literally just the order of the amino acids. That's it. When we talk about primary structure, especially when we order them, it's assumed that a peptide bond has been formed between all of the amino acids. So between the C terminus of anything that comes previous connected to the N terminus of anything that comes after an amino acid. So it's a sequence of these peptide bonds or these sequence of these amide bonds. And we've already gone through this exercise of showing, well, what's the chemical reaction? We have a carboxylic acid, reacts with an amine, and then it forms an amide or a peptide bond in the sense of a protein or a peptide. And so I've already mentioned this, but a protein is a polypeptide, poly being many, peptide being amino acids connected through amide bonds of 50 or more amino acids. And specifically if they have biological activity, though you can expand that definition to pretty much any amino acid. It doesn't necessarily have to have biological activity, but if it's made by the body, you assume that it's going to have biological activity. And so the primary structure, and the way we usually write primary, and we've seen this a couple times now with alcohols and amides, you might see one with the degree sign means primary. So if a protein is the particular sequence of amino acids held together by peptide bonds, so it's just the sequence, the order that they appear, that's it. So what I have shown on the bottom is a peptide made out of 4 amino acids, right? We have alanine, leucine, cysteine, and methionine. And so actually it looks like a lot is going on at once, but if we stop and slow down, we can start to pull apart individual amino acids. We know that each individual amino acid is going to be separated by an amide bond. So if I go through and look at my amide bonds, I'm going to notice I have one here, and one here, right? We also know that we should have an N terminus and a C terminus, and we do. Other things that we can start to pick out. Alanine has a functional group of just a methyl group. Leucine, we know, is nonpolar, and that's the structure of leucine. Cysteine has that thiol group, so there's cysteine's R group. And then we have methionine, which is nonpolar, but also has that sulfur kind of stuck in the middle. The reason particularly why it's nonpolar, because you can see it's symmetrical, almost symmetrical on both sides. But we can slowly start to dissect what's going on. So we can see we have, if I start breaking things up theoretically at the amide bonds, well, there's my alanine. There's my leucine. There's my cysteine, and ends with a methionine. And so what I'm going to do in the next slide is show you how to take these explicit structures, which actually looks like there's a lot going on, because there is, but using skeletal structures will help simplify visually what's going on. So why don't I take that same polypeptide, right? We said it's going to be, and I'm going to use the one-letter codes. We said it's going to be alanine, leucine, cysteine, oops, I'm thinking serine, sorry, cysteine and methionine. So if I go through in a very, very ordered way, I know all proteins, at least by convention, we start with the N-terminus. So we know we're going to have an amide or an amine group, right? It's protonated because amino acids in the body are always surrounded by water. We have an alpha carbon that's connected to an R group. The R group for alanine is going to be a methyl group. That's connected to the carboxy terminus. But we know in a polypeptide, the carboxy terminus becomes an amide. That's also connected to the next alpha carbon in the next amino acid, which happens to be leucine. And if I draw it as a skeletal structure, will look like that. Sorry, there's a couple of gaps in there, but you can see what's going on. That's connected to the carboxy terminus of the leucine, but it's part of a peptide. So it's not going to be a carboxylic acid, but another amide, which is connected to the next alpha carbon, which is cysteine, which has that thiol group. That's also connected again to its C terminus. It's not done yet. So we know the C terminus is not the end, at least in this case. So it's an amide because it's connected to the last amino acid, which is methionine. That's connected to the next alpha carbon, which is methionine. And if I draw methionine out, we have that structure. So it's a little bit, we can see the zigzag. We have 2 carbons, 1, 2, sulfur, and then we have that methyl group on the end. And then for methionine, because it is the true C terminus, it will be a carboxylate. So it takes practice going through, but we can see that it's more cleared up, at least in the way I see it. And again, for the exam, if it's more easy for you to write explicit structures, as long as you stay all explicit, you'll be fine. If you like dealing with skeletal structures, which I think is easier to see, that's my opinion, stick with all skeletal. Don't mix and match. But we have our alanine, we have our leucine, we have our our cysteine, and then our methionine, right? So again, practice in writing the skeletal structures of amino acids, specifically in this example of peptides. So to give you an example, right, why is primary structure important? So we're going to stick with small peptides, not necessarily a protein, but it says a thyroid hormone that stimulates the release of thyroxine is a tripeptide with the amino acid sequence glutamine, histidine, and proline. Although other amino acid sequences of these 3 amino acids are possible, right? You could have pro-glu-his, his-glu-pro, or whatever combination you want of the 3. But only this specific sequence, or if you want to put it into a different parlance, right? Only this specific primary structure of glutamate, histidine, proline actually produces hormonal activity. The reason for that is your body only recognizes that sequence. If histidine comes before glutamate, right, you're not going to get that activity. So your body is fine-tuned to these particular sequences. Not necessarily in the sense that it's reading things linearly, and we'll get to this when we get to secondary, tertiary, and quaternary structures, but it's how all those atoms are placed in space. That's usually where the biological activity comes from. So an example, and we've already looked at this when I showed you from the protein database, when we looked at those 3D structures, but we look at the primary structure of insulin. And so what I have on the right-hand side, right, so insulin is actually made out of 2 separate chains of amino acids. So technically each is a polypeptide chain, but when your body is making them, it will make an alpha chain or an A chain and a B chain. Those are going to be linked through disulfide bonds. We've already seen disulfide bonds, I think it was chapter 15. It's when 2 thiol groups that are near each other are oxidized, and so they form a covalent bond between the 2. And that's what you're seeing in those 2 highlighted groups I have in between the 2 chains. So technically they're separate polypeptides, but the way that it's functional in your body, they need to be covalently bonded together. There's a third disulfide bond in there. The body usually uses disulfide bonds, especially when they've been excreted outside the cell, to keep the structure of the protein. We won't get into that, but inside cells, disulfide bonds tend to be reduced, which means they'll be formed, they won't form, which they'll either, they need to be protected, so they'll be inside the protein, so those are fine. But things that are on the outside would definitely be reduced back to thiols. Excreted outside cells, disulfide bonds tend to stay intact. And so insulin travels throughout the body, they're outside the cells, those disulfide bonds will usually remain intact, right? But take home message, we actually have two distinct chains. Each of those is a primary structure. So I can maybe list off the first 5 amino acids, right? You look at the A chain, we have glycine, isoleucine, valine, glutamate, and glutamine cysteine. We look at the B chain, we have phenylalanine, valine, asparagine, glutamine, histidine, and leucine, right? It's the order in which these amino acids appear. So if we have that under our belt, we can start to move into the other levels of structure. So we have secondary, tertiary, and quaternary. That literally means, so we start with primary means first, secondary means second, tertiary means third, and quaternary means fourth level of structure. And so we'll start off talking about secondary, and then we'll build ourselves up. And I'll bring in examples with 3D structures as we go along. So these next 2 slides, they're not super clear. I'm doing the best that I can, but I'll switch to talking, or showing you rather, through the protein data bank, what I mean by what does the secondary structure look like. So we're gonna look at 2 levels of secondary structure. There's technically 3. The third is random coil, which means there is no structure. So either you have no structure, so random coil, or we're going to have 2 very ordered structures. The first is an alpha helix. The second will be a beta sheet or beta pleated sheet. And there's 2 types of beta sheets. The book doesn't get into that. We're just going to call them beta sheets in general. So secondary structure, what that means is, depending on the order of the amino acids, there tends to be, and we've seen this as experimentally verified, there tends to be sequences that will form larger structures. And what do I mean by that? The first type of structure is a helix. So similar to DNA, it's not a double helix, it's just a helix. What ends up happening is these amino acids start to spiral upwards. And if we look at the backbone of an amide group, right? If I look at, let me just draw quickly, just, I don't know, we'll make it alanine, right? And then I'll connect it to something. Let's make it, I don't know, cysteine again. Right, the idea is as these molecules start spiraling upwards, they actually start to stack on each other. And there's going to be hydrogen bonding between the carboxy group that's going to be found in the amide backbone or in that C terminus, right? And the NH group of each of the amides. So it's hard to see when I only have 2 of them in a row. Not super easy to see here in this figure, but the idea is that these amino acids are spiraling upwards. And as you spiral upwards, you could have a carboxy terminus making a hydrogen bond with a lower amide group. And that's what you keep seeing through this figure here. And those hydrogen bonds are actually giving structure to this, this secondary structure. And helices are very common to find in proteins. There's a lot more to it rather than being structural. It affects dipoles. It affects the dipole moments of metals. It affects the redox potential of metals. They have a lot of things going on, but for us, we just care about structural. So what I want to show you first is going to be, let me get out of notation mode. I'm going to show you hemoglobin. So let me do that. And so hemoglobin, and we're going to pull this apart slowly, right? Hemoglobin, most people know from biology in high school that it's responsible for binding oxygen, right? And that transports it through the red blood cells throughout the body, delivering oxygens to other cells. Couple of things right off the bat. Number one, you see that there's 4 different colors on the screen. That's actually the tertiary, the quaternary structure. That means how many bits and pieces of different proteins come together to make one protein. We'll get there, right? What I really care about is when I move this around, we've already talked the difference between cartoon representations of proteins and then also the explicit structures, which are really not confusing, but there's a lot going on because you have hundreds of atoms. But what you see here, right, we can definitely see in this secondary structure, well, these random bits, you see how they're not really structured? Those are random coils. We're not concerned about those. But you see there's a tremendous amount of alpha helices, and they give it the structure necessary for hemoglobin to do its function. Also, what you might see, and maybe you did learn this in high school as well, is that in areas like around here, this is actually the heme group. It's porphyrin or polyporphyrin, I forget, I should know that. I think it's polyporphyrin, right? In the center, you can actually see the iron atom right here, that orange ball in the center in the heme group. That's where oxygen binds, right? And actually, out of this whole giant structure, I know there's a lot going on, there are only 4 heme groups. Each one of these giant structures only carry 4 molecules of oxygen. So you can imagine how many molecules of hemoglobin you have in your body at any given moment. But again, what I'm trying to show on this one is we're looking at the secondary structure. So you can definitely see the helices. So we can definitely see down a barrel here, but see that it's a helical structure when we look at it on its side. So let me go back to the slides and we'll keep moving on. And so that's what I'm looking for in terms of alpha helices and secondary structure. The third type of secondary structure, remember I said random coil, alpha helix, is going to be beta sheets. So it's beta pleated because you really can't see it in this figure, and it's actually kind of hard to see even in the crystal structures. The shape of the beta sheets actually does look pleated, where it looks like there's ridges in it. You really can't see it, but you can kind of imagine there's ridges there. If you get really into the structure of the protein, you can see it. I'm not so worried about that. But again, there's way that beta sheets function as structural units. The way in this one is instead of making helices, where those amide backbones and those, the carbonyl oxygens and then the amide hydrogens form those alpha helices, there are certain primary structures that tend to form beta sheets in which they actually wrap around on themselves. And you can see there's a hydrogen bond, hydrogen bond, hydrogen bond, hydrogen bond. They're holding them together, right, in such a structure. And these structures have a huge role in the function of the protein. And so what I want to do is pull up another type of protein that relies heavily on beta sheets to keep its structure and its function. So we saw previously, sorry, I lost my cursor, there we go, that the structure of hemoglobin was dependent upon so many alpha helices. So what I want to look at now, and I got to wait until, there we go, my Zoom display moves away. We're going to start looking at something called green fluorescent protein. So these are proteins from jellyfish. So have you ever seen anything in nature glow green, or any biological, I'm not talking about glow sticks, that's a very different chemistry. Well, actually the chemistry is kind of similar on a physics level, different in terms of chemistry, but you get a green glow from these proteins. We're not going to get into this. When you crystallize proteins and you shoot X-rays at them and get crystals out, or crystal structures, information on how the amino acids are arranged in space, occasionally you get weird shaped crystals, and this is one of them. So a crystal is just a repeating unit. The repeating unit in this particular crystal just happens to have 2 of the GFP, green fluorescent proteins, crystallized in a repeating unit. So it's technically not a quaternary structure, and maybe it doesn't make sense when I say that, but it will when I get to the further slides. But if we just look at one of them, right, you can see these wrapping beta sheets literally make a barrel. And they're actually, this giant structure is usually called a beta barrel. And it's hard to see, but maybe you can see a little bit, like we can see some of the structure right here that's highlighted. This is actually called something called a fluorophore. When it's hit with certain, or when any kind of energy is given to it, it has to be specific, but it can excite it and it actually emits light, and it emits green light. Again, I said these are both actually identical. It's just an artifact of crystallization, but the main point I'm looking at here, though, you can see the beta sheets are making a very distinct structure. And actually, the thing that makes this protein special is those beta sheets are arranged in such a way that mostly water is excluded from the inside. So it's actually pretty dry on the inside. So it helps that fluorophore actually do its chemistry to make light. So that's going to end our discussion on secondary structure. Then we'll talk really briefly about tertiary, and then even probably more briefly about quaternary. So a tertiary structure is going to be the overall three-dimensional shape. So even though we looked at hemoglobin and then we just looked at that TIM barrel, is it a TIM barrel? The beta barrel of the green fluorescent protein, right? Only each one of those spirals, or each one of those, what it looked like when we were looking at the GFP, right? It was just like one little strip. When everything came together, that's when you saw that three-dimensional shape, right? That three-dimensional shape overall is actually giving it its function. So what I'll do is I'm going to go back for a second after I'm done reading this slide to distinguish between secondary and tertiary structure in both of those examples of hemoglobin and GFP. One thing before going into that, and we're going to have another slide on this in a second, the book uses the term cross-linking to talk about how secondary structures come together to hold a protein together into like a three-dimensional shape. I'm hesitant to use that word because I've never heard a biochemist use the word cross-link without being a covalent bond. And hardly, it's not uncommon, obviously, because disulfide bonds are covalent. Most of the interactions that keep a protein together aren't covalent. They're going to be intermolecular forces, and we'll talk about the types of intermolecular forces, I think, in the next slide. Just be careful with terminology. Typically, when I say cross-link, or I've ever heard a scientist say cross-link, it's a covalent bond, not a non-covalent bond. So that's just my own little caveat. But let's go back to those two structures, right? So we can start first with the GFP. So one of these things, so if we look here, right, the one I have highlighted kind of in pink, this one strip, technically with the one running anti-parallel to it right here, we're not getting into that, but technically that is the secondary structure, is what I'm looking at. The tertiary structure is everything. How do these individual pieces of secondary structure, so each beta sheet, each random coil, how do they come together to make this giant barrel-shaped protein? So secondary structure is just the beta sheets. Tertiary structure is the whole structure, right? And again, I said for GFP, it's an artifact of crystallization. There's two of them. But each individual one inside the cell operates by itself. When we look at hemoglobin, right, one piece of secondary structure would be like this helice right here, this helix right here, right? That's a piece of secondary structure, or this helix here, or that helix there, right? But together, like this orange thing, that is the tertiary structure. This magenta thing, that's tertiary structure. And as foreshadowing, you can see, unlike the last GFP example where it says an artifact where two different things were crystallized together, it's been shown that hemoglobin only operates if four of these are together. So each one of these is called a subunit. So it's literally the unit, but what are the pieces that make up hemoglobin? So there would be a green unit, a blue unit, an orange unit, and a magenta unit, right? So there'd be four subunits. and they only function when all four of them are together. And they are not covalently bonded. And we'll talk about that in the next slide. Everything in this figure you see here, other than this heme group, which is covalently bonded, which means it's making a physical bond, everything else is held together through intermolecular forces. So why don't we take a look at those? And so the intermolecular forces that keep proteins together, right? This is kind of like a generic representative figure saying the whole thing's tertiary. But remember, I was talking about individual units, like that's a unit of secondary structure. This beta sheet right here, that's a unit of secondary structure. The whole picture, this whole thing is the tertiary structure. So again, remember, we're going up in terms of complexity. So things that hold proteins together. I don't like the way the book does this, but then again, we might be getting into a little bit too deep into this, but we're looking at hydrophobic interactions, right? Those are between the non-polar amino acids. So what you see here are two phenylalanine groups in close proximity. What you have here are two methyl groups. Both of those groups, phenylalanine and methyl groups of alanine, are hydrophobic. Remember, they're water hating. Those tend to actually form the inside of proteins because they want to avoid water. And that's actually what gives majority of the structure to proteins is hydrophobic interactions. Other interactions help kind of staple things together that are on the outside to give the, the form of the protein that actually does the interacting with everything on the outside. Hydrophobic kind of gives it that core structure. So other type of interactions that hold things together are going to be like hydrogen bonds. And we saw this when we were talking about both beta sheets and alpha helices, but there's a lot of electronegative elements in proteins. So let me see right here, we have a carboxy terminus. Maybe it's going to interact with the N terminus, right? You can have 100 amino acids, and actually it's very common for the end of a protein to come back and just interact with its beginning because they're attracted to each other. Doesn't always happen, but it can. And that happens a lot inside proteins as well. They tend to be weaker bonds. I mean, they're strong, but they're not as strong as covalent bonds. And that's why proteins, when you heat them up, start to fall apart. And we'll talk a little bit about that too. Again, hydrogen bonds don't need to be between the N and the C terminus. Like you could have something here from serine with another serine, can definitely link them together through a hydrogen bond. Hydrogen bonds between beta pleated sheets, right? Again, a lot of these interactions going on. A salt bridge is going to form between something, say like aspartate, which we have here, which is an acidic amino acid, could interact with, I don't know what that is. I'm going to make a giant assumption that it's a lysine because it's not showing anything but the last amino group. And the only amino acid that ends like that is going to be lysine, right? So it could be a lysine interacting with aspartate. You have a negative and positive charge. They're attracted. That's strong. And then you also have those covalent bonds that they act as staples. And these are not going to break unless you do specific chemistry on them. Disulfide bonds can make parts of proteins just staple together and they'll never move. And so what most people think of as proteins as not moving, like they're just like their structures and they float around, they're actually constantly vibrating. They're constantly breaking their structure, coming back together. They're constantly moving. So it's almost like having a jittery 3-year-old. It's kind of like when we think of like protein structure. They're just moving around constantly, but they have a general shape. And they usually can only do their structure or function when they're in this general shape. So that's why nature has evolved certain ways to try and keep these shapes together. But occasionally they break it and they just fall apart, but then they'll reform. And it's happening very, very quickly. You're talking maybe milliseconds, nanoseconds, things like that of coming together and falling apart and coming together. So they are constantly moving. And so here's just a list of everything that I just mentioned. So maybe you might want to pause this later. I'm trying to, I've realized I'm slowing down, so I want to go faster for time. Come back. I do expect you to know all of these interactions and the type of amino acids that make these interactions. So as an example of why things might go wrong when proteins don't have the correct primary structure, because if you mess up the primary structure, You can mess up the secondary structure. If you mess up the secondary structure, you mess up the tertiary structure. You mess up the tertiary structure, you can sometimes mess up the quaternary structure. And so this is how mutations can actually affect the body profoundly. And so an example of this would be sickle cell anemia. So sickle cell is a mutation in hemoglobin, so that one of the proteins that I showed you, and it's literally called a point mutation, and we'll get into this when we talk about in chapter 17. A glutamate due to a mutation in the DNA. So DNA is responsible for giving the instructions to make proteins, and we'll talk about that and how this happens. But glutamate was mutated to valine. So you went from polar acidic, which has one particular, it can make hydrogen bonds, it can make salt bridges, to something that can do neither. All it is nonpolar. And actually, this profoundly affects the structure of hemoglobin so much that it actually causes a disease from a single mutation. A regular red blood cell is circular, and it kind of looks like if you put an indent in the center of it. So it's kind of like donut-shaped-ish without a hole through the center. Red blood cells don't even contain DNA, I believe. Yeah, they don't. They just are just full of hemoglobin. So when you start making mutations to the hemoglobin, they actually have this sickle shape to them, and it starts changing their structure, and they can't do their job as well. So it clogs capillaries. Once that happens, it causes inflammation because your body knows something's wrong. That causes pain and organ damage, and it's not as effective at delivering oxygen, all from messing up one amino acid. So let's get into the last part. There's actually one more slide after this that I want to talk about. What happens when I break down all these structure, but quaternary structure. And I'm not going to show the figure again because it's actually shown here in a more cartoony form than they are the cartoon form, but we're looking at hemoglobin, the same structure I showed you from the protein data bank, right? So quaternary structure is the combination of two or more protein units. So each is called a subunit. So in the picture that I showed you from the protein data bank, the PDB, right, there are 4 different colors. Here, they're only using 2, but you can see they're alternating, right? But it's 4 polypeptide chains make our subunits in a single massive molecule of hemoglobin, right? They stabilize each other and they actually interact with each other. And that actually as one molecule of oxygen binds to one subunit, it makes it easier to bind to the second. Those 2 make it even easier to bind to the 4th. And after that, it makes it even easier to bind to the 3rd, then even easier to bind to the 4th, right? So they actually work together. We're not going to get into allostery. Allosterics means how protein subunits talk to each other. That's beyond the course, but they do talk to each other. We call it crosstalk. They talk to each other, not in words, but through interactions. So here's just a summary slide of, going from primary all up to quaternary. So primary is just the order of amino acids. Secondary is alpha helices, beta sheets, or random coils. Tertiary is how they all come together in space and how they're held together with either hydrophobic interactions, salt bridges, H bonds, or disulfide bonds, right? Most proteins, I wouldn't say most, I don't even know what the percentage would be. A lot of proteins stop at tertiary, but a lot of proteins also form higher level complexes called quaternary structures, where multiple of the same subunits come together to make one giant functioning protein. So we have our primary, second, tertiary, and quaternary. So in the project that you might have noticed, so I'll make a separate video on that. So I'm not going to make this any longer than it has to be. So I'll make a separate video about the project. In particular, some COVID, the SARS-CoV-2 proteins have quaternary structure. Some only stop at tertiary. I just want to clarify that later. Last slide, then we'll be done. Then I'll make another video on enzymes. So what happens if I mess up either the tertiary or the quaternary structures of proteins? There's multiple ways I can do that. Some proteins can actually snap back and just, it doesn't matter if you mess with them and you put them back into the situation or the environment that they like, they might just snap back and have no problem and go back to doing whatever they were doing. Other times they actually die, right? They're effectively dead. They don't do what they're supposed to do. They're messed up now. The body usually eats them and then recycles them. So there's a couple of ways to commonly denature protein. One is to add heat, And that makes sense. You cook an egg, the egg is mostly protein. You heat it up, you mess up the tertiary structure of the egg proteins. They turn into a giant jumbled mess, and you have, you eat it, right? Organic compounds can also mess with the structure of proteins. So when you're talking about, this is not always the case. You need high levels of alcohol for this. But when you do use like ethyl alcohol or isopropanol to sterilize things, a lot of the times it is denaturing the proteins on the outside of bacteria. What it's doing to viruses, specifically COVID-19, it actually tears apart it's surrounded in lipids, and then alcohol is really good at tearing those apart. That's why that happens. It's not necessarily a protein thing. Acids and bases can break amide bonds. We didn't talk too much about that. Well, we did actually. I did give you an example in the organic chemistry, the organic homework practice problem, number 68, I believe it was. You saw what happens when an amide bond is attacked by a base, right? It breaks it. That's exactly why acids and bases hurt you. When you touch an acid or base, it's actually breaking, one, it's breaking the fat in your skin, but it's also breaking proteins in your skins irreversibly. Acids can be reversed a little bit. Bases actually are very destructive to protein structure. It literally snaps protein bonds. Heavy metals, when you ingest lead or mercury, these metals will actually bind to cysteine, and then it messes up the protein because it's going to affect their tertiary structure. It can also set off immune responses because your body realizes something's off. And so sometimes you can have an allergic reaction. And then also agitation. Proteins are weakly held together. I've had protein solutions that have dropped on the floor and they're dead because they're just full of foam because they all died. Why? Because what ends up happening is because they're held together, but not covalently, you shear them, you shake them up or something like that. You can rip apart the protein on that level with just shaking them, right? So when you make whipped cream, you're whipping that up. You're breaking all the proteins that causes whipped cream to form. You're stretching and breaking peptide chains, and sometimes you can actually break them. Though that usually, the book says you can break them. That takes a lot of force. Mostly what you're doing is breaking the non-covalent interactions when you agitate them. So the example I gave was like cooking an egg. Exactly. You have albumin or lysis. Is it albumin? Actually, no, not 100% sure actually what egg is made out of. So don't quote me on that one. But you do have egg protein, or egg is very high in protein. It has a very particular structure, right? It's very liquidy. It moves around. You heat it up, or you applied acid. If you ever actually even add like vinegar to eggs, you ever heard of like cooking fish with acid? You're not cooking it. You're just denaturing the proteins, right? You're not doing it with heat. But you can mess up the structure. You make it denatured. It just basically starts forming a non-useful structure, and they kind of stick to each other. And that's why cooked eggs look solid versus liquidy, is because it just becomes a jumbled mess and they all stick to each other. So again, here's some protein denaturations. The book gives you like this cutoff at 50 degrees C. That's a generic cutoff. Thermophilic proteins don't care about 50 degrees C. Proteins in your body definitely will die at 50. There's plenty of proteins that don't degrade at 50 degrees C, but they're just making a generic number. That's all. All right, that was long enough for that one. Sorry, that was a little bit long. It's the stuff that I love to talk about, but this is an important chapter. I want to make sure we do cover it fully because I think in terms of the biochemistry chapter, this one will probably serve you the most in the career that you guys have chosen. All right, so I'm going to end the video on this. I'm going to make a short video on enzymes after this, and that will be the end of chapter 16.